Welcome back, fellow mitochondriacs, for another episode of Cancer as a Mitochondrial Metabolic Disease. I'm happy to be back with you today to share, kind of, at least as for right now, one of the last strategies that I've been able to come up with to help you lower your glucose-ketone index, to help you lower the available glucose fuel to tumor cells, to get you into maximum ketosis and to help you with obviously your goal of becoming NED with whatever disease process you have. Today, we're going to be talking about cold, and I'll be very frank with you. I think that cold is extremely promising. What I also think is that cold is extremely under-researched, and I'm hoping that that will change in kind of the near future as now, cold plunging and things like that have become more mainstream and more kind of accepted and it's kind of a fad, I would say, right now. So therefore, hopefully that means that dollars will fund and go into research, and there'll be an appetite to kind of get this information, because I think it could be very, very useful for our purposes and for other purposes as well. So let me, without further ado, get into this PowerPoint. So I'm going to start out by talking about mitochondria's effects when exposed to cold. And what I'll say is that I'm going to go back to some graphics that we have not looked at for quite some time, probably like last summer, summer of 2024, because I had kind of a little bit of a micro series on the mitochondrial supercomplexes and mitochondrial performance, and explaining kind of why that's important. And I think it deserves a refresher, given that the first slide we're talking about cold, we're talking about mitochondrial cristae formation. So before I read off this slide, I just want to reiterate, remind us that in biology, generally, structure equals function. So if the structure of something is off or broken, then generally it's dysfunctional or completely non-functional. So the mitochondria are no different from that. So the shape of the mitochondria, how long they are, how short they are, how organized when it comes to their inner membrane folds called the cristae formation, all have very important effects on how functional that mitochondria is. And so this paper is titled Mitochondrial Cristae Shape Determines Respiratory Chain Supercomplex Assembly and Respiratory Efficiency. And I pulled this next graphic because I wanted to show that when these mitochondrial proteins, electron transport chain proteins, are in the proper configuration and they're assembled into supercomplexes, or the respirosome, as they're also known as, then they are much more efficient at producing ATP. And when they're disassembled, then they don't produce as much ATP. They produce a lot more reactive oxygen species, and that becomes a vicious cycle, as we talked about in the past. So when we have any kind of pretty much any time, I'll back up. I'll say that the super complex and the respirosome concept is fairly new to biology. We're talking like 10 to 12 years old, and it wasn't even solved, the structure of it, until like 2017, 2018. So it's a very new kind of part of biology, and they're still trying to understand all the facets of how it assembles and what the factors are, and it's very fascinating. But I say that because a lot of times in the literature, when you're looking through things that improve mitochondrial function, they may not say improves mitochondrial supercomplex formation, but what they'll say is they'll say improves cristae formation. And then that since the cristae determine the supercomplex formation and efficiency of the respiratory proteins, you know that if something says that it improves crystal formation, that it's going to improve the mitochondrial supercomplex formation, ATP production, etc. And I give that backstory because the first slide I picked was a paper showing that when we are exposed to cold, it improves our supercomplex formation. So this paper was published in Cell Metabolism, Cell 2021, and it says, and the title is a cold stress-induced PERK OGT axis-controlled TOM70 assisted mitochondrial protein import and cristae formation. And what I highlighted here at the bottom was that we have identified a cold stress interorganelle axis that increases cell respiration through mitochondrial protein import and subsequent cristae formation. These studies have significant implications on the bioenergetics and adaptations to cold stress. So I say that because I like to give a mechanism as of how it's going to affect us clinically down the line. So the first way I want to talk about a clinical outcome, okay? Like we know this is important in kind of a vacuum. We know mitochondria are important. We know mitochondria are what are responsible for making the vast majority of our energy in our healthy cells. And we know they need to be healthy and functioning adequately. But what does that actually translate to clinical outcomes? You know, stepping out from the bench to the bedside, what happens, right? Well, that's where I would say there needs to be a lot more work done. But I think what we have is very promising. So this is a paper that I pulled from the Journal of Applied Physiology in 1985. And it says here, the examining of benefits of cold exposure as a therapeutic strategy for obesity and type 2 diabetes. And it says here that the pathogenesis of metabolic diseases such as obesity and type 2 diabetes are characterized by a progressive dysregulation and energy partitioning, often leading to end-organ complications. One emerging approach proposed to target the metabolic dysregulation is the application of mild cold exposure. In healthy individuals, cold exposure can increase energy expenditure and whole body glucose and fatty acid utilization. Repeated exposures can lower fasting glucose, and insulin levels, and improve dietary fatty acid handling, even in healthy individuals. Despite, I'm gonna take a break for a second. So one of the things is that most of the papers done on insulin resistance, diabetes, blood sugar, fasting blood sugar, looking at people's A1Cs, are participants that are diabetics or pre-diabetics, which makes sense because that's who they're trying to treat. But it would be nice to know that, let's say hypothetically you didn't have cancer. Is this still going to have a very profound effect on me when I'm adding this into my kind of regimen for enhancing metabolic therapy? And at least in some glimpses, we get some information about that. Repeated exposures can lower fasting blood glucose. Okay, we saw that. Despite its apparent therapeutic potential, little is known regarding the effects of cold exposure in populations for which this stimulation could benefit the most. The few studies available have shown that both acute and repeated exposures to the cold can improve insulin sensitivity and reduce fasting blood glucose in individuals with type 2 diabetes. However, critical gaps remain in understanding the prolonged effects of repeated cold exposure on glucose regulation and whole body insulin sensitivity in individuals with metabolic syndrome. Much of the metabolic benefits appear to be attributed to the recruitment of shivering skeletal muscle. However, further work is required to determine whether the broad recruitment of skeletal muscles observed during cold exposures can confer metabolic benefits that surpass what has been historically observed from endurance exercise. In addition, although cold exposure offers a unique cardiovascular responses for physiologic stimulus that increased energy expenditure, further work is required to determine how acute and repeated cold exposure can impact cardiovascular responses and myocardial function across broader scope of individuals. So what are they saying? they're saying, okay, well, they know that at least part of it works because of the shivering response you get from the skeletal muscles. And that's going to increase caloric demand on those organs. So therefore, they want to know, number one, are there any additional, mechanisms that can help elucidate why are there these, blood glucose lowering, insulin, improving responses in these either healthy or, diabetic patients. And, basically, is there any benefit to cold over just normal exercise? And I think those are good questions to ask. So the next graphic I want to show was that they break down these responses by metabolic responses and cardiovascular responses. And they give a bunch of different data, basically for mild cold versus moderate cold versus high intensity cold. But I wanted to highlight in particular on this graphic, this portion right here, that mild cold exposure will increase your resting metabolic rate, your metabolic rate by 1 to 2x. Moderate cold, 2 to 3x, and high intensity cold, 3 to 5x. So that's pretty unbelievable, really. And we're going to see in some of these later papers what that potentially could look like for us. It later says that whether the cold stress is uncompensatable, fall in core body temperature, or compensatable, no change in core body temperature, the relative increase in metabolic rate above resting times RMR confers commonly referred as cold-induced thermogenesis. The reason I particularly highlight this is because one of my mentors, Dr. Jack Kruse, talks about cold thermogenesis, cold thermogenesis. What is cold thermogenesis? Cold thermogenesis is a progressive increase in our metabolic rate when we're exposed to cold. And in particular, as we'll see later, there is a high degree of increased fat and glucose burning that when that is done. And it's believed to be from a shivering response. There's also brown adipose tissue that's involved as well as increase in mitochondrial performance and an enhancement of oxygen consumption as well. So there's a lot of different mechanisms kind of going on with this. But I just thought that was very unbelievable that a mild cold stress can increase your metabolism by 2x, moderate up to 3x, and high intensity up to 5x. I just thought that was unbelievable. We're talking about 500% increase in your resting metabolic rate when you're exposed to cold. It later says that what has become clear over several decades of cold exposure research is that under fasted conditions, carbohydrates, lipids, and proteins can all provide the necessary substrate to fuel thermogenesis. So basically, our healthy cells can use whatever means necessary, carbs, proteins, fats, in order to fuel this cold-induced increases in our metabolism, or thermogenesis. Indeed, when one fuel source is depleted or restricted, the others can compensate to maintain ATP production, shivering activity, and whole body thermogenesis. So therefore, if you were to happen to be on a low-fat, low-protein diet, your body would use glucose exclusively to maintain the shivering activity. If, on the other hand, you are on a ketogenic metabolic diet and you are only basically doing fat and some protein, your body would, the healthy cells would adapt and they would use that as the source. However, it says later, carbohydrates appear to be the most important of these fuels. Although carbohydrates account for about 1% of total energy stores, its relative contribution to total heat production increases in parallel with increases in cold-induced thermogenesis, accounting for up to 80% of heat production at the highest shivering intensities, whereas fatty acid oxidation increases in response to cold but remains steady at about 100 to 200 milligrams per minute, regardless of the shivering intensity, sex, carbohydrate supplementation, or even the suppression of white adipose lipolysis. So what is that saying? It's saying that even though these cells can use glucose, protein, or fat, it seems to prefer glucose over everything else, which could be, again, very beneficial for our purposes because we want glucose to be maximally used over other tissue, other fuels to starve the cancer, essentially, right? And it says here, when given a standard meal or a carbohydrate-rich beverage during a mild cold exposure, the rate of fatty acid oxidation, fatty acids burned, fat burned, was 37%. Even if you're giving carbs at the same time, that's pretty unbelievable because generally when we induce an insulin response, that shuts down our carbohydrate burning. So we're increasing fatty acid burning by 37% when given a meal or a carbohydrate-rich beverage. But get this, but carbohydrate utilization increased by approximately threefold to as much as 6.6-fold. So you're getting a 300 to 660% increase utilization in carbohydrates or sugar consumption when you're exposed to cold. That's unbelievable. And that's really beneficial for our purposes in particular, using cancer as a metabolic disease and suppressing our glucose through diet as much as we possibly can. This dramatic increase in glucose utilization combined with the suppression of endogenous glucose production results in a no net change in glycemia under ingestion. So basically what they're saying is because it suppresses our endogenous carbohydrate production and it completely like uses glucose 300 to 600% more than normal. If you were to drink a high carbohydrate beverage, they're seeing no change in your blood sugar whatsoever, which means you're completely mitigating any carbohydrates you're putting in your body when exposed to cold. I think this is really important information. So let's look at a really neat graphic that it showed as well of kind of like multiple organ systems that are helped when exposed to cold. So there is neurotransmitter function increase. It has a question mark next to it, but I've seen some other studies showing that it has a pretty profound increase of norepinephrine, one of our main neurotransmitters in our brain. Decreased glucagon secretion, decreased insulin concentration, so it suppresses insulin secretion. Increased cardiac output and increased blood pressure, increased stroke volume while you're under cold because you're trying to circulate blood more, of course. You're under the influence of catecholamines, which is going to do that for a short period of time. Improved hepatic insulin sensitivity, triglyceride production or storage, global inflammation, AMPK, adipose tissue inflammation, improved insulin sensitivity at the level of the muscle cells, increased thermogenesis, decreased glucose. Let's see, what does it say? Increased glucose uptake, reduced total cholesterol, reduced LDL or the bad cholesterol, quote unquote, and other kind of hormone signaling with adiponectin, leptin, et cetera, et cetera. So it seems like there's a whole body improvement that happens when you're exposed to cold. Now, I'll be the first one to tell you that I hate cold. I moved to Florida for a very particular reason because I hate being cold. But I can tell you that this research, along with other research like this mitochondrial research, has led me to very likely get a cold plunge in the next 3 months, because I think that this could be part of a very good preventive medicine practice as I get older. But I do generally recommend for people or clients who have the ability to either have a local cold plunge facility, whether they have the funds to purchase their own cold plunge, or if they live in a northern latitude where cold is already present, doing a cold, bath like in a natural water or something like that, without hurting yourself, could be a beneficial practice. I think that, potentially, swimming in cold water or something like that, getting the exercise component could give us a double or triple whammy probably as well. So there's a lot of things that could be done with cold. Let's keep going. So this is another paper that was published, again, International Journal of Sports Medicine in 1992, energy metabolism during cold exposure. And I highlighted here, whereas previous human studies have concentrated on cold-induced changes in metabolic levels of hormones, and metabolic substrate, recent work from our laboratory has demonstrated that cold exposure to cold at rest, shifts substrate utilization from mainly lipids at thermal neutrality to carbohydrates, representing the main fuel for shivering thermogenesis. Further investigations have revealed that the marked increment in carbohydrate oxidation in cold exposed humans is derived from a greater utilization of both circulating glucose and intramuscular glycogen stores. With respect to lipid metabolism, recent studies have shown that cold-induced increase in lipid oxidation in man is fueled primarily by fatty acids released from white adipose tissue, triglycerides, other possible intramuscular triglycerides, not plasma triglycerides. So I guess looking at cold exposure from a non-cancer related perspective, it seems that even though we don't have robust trials on cold's ability for us to have us lose weight, it makes a ton of sense that it would. And I really hope that this catches some steam within the research community, because when I read that it can increase your carbohydrate consumption up to 600% and fatty acid production at up to 37% while given a high carbohydrate meal and leading to no glycemic changes, like there's no change in your glycemic index or your glycemic or blood glucose when you're given a high carbohydrate beverage when you're exposed to cold, it seems like this could be like almost like a cheat code to dieting and weight loss. Again, there needs to be a lot of research done for this before we're ready to, before it's, ready to make broad claims and things that we can't back up. But it just seems that it just seems to be an unbelievable modality for general health, mitochondrial improvements, et cetera, glycemic, improving your glycemic control, although we still have a lot of work to do from that standpoint as well. Moving on here. So this is a more recent paper that was published in 2023. And it says here, and the paper is titled Physiological and Molecular Mechanisms of Cold-Induced Improvements in Glucose Homeostasis in Humans Beyond Brown Adipose Tissue. And it says here, hence, the exact tissues and underlying mechanisms that mediate cold-induced improvements in glucose homeostasis in humans remain to be fully established. Now, mind you, the first paper we looked at was from 1985. So we're talking about a long time of research, but unfortunately, there's very few papers or very little research has actually been done. So it's just really sad that even in 2023, there was very little understanding, or not very little understanding, but there is an incomplete understanding of how cold, something as simple as being in the cold, water or cold, temperatures can induce improvements in glucose homeostasis, but I'll digress. In this review, we evaluated the response of the main organs involved in glucose metabolism, pancreas, liver, white adipose tissue, skeletal muscle, to cold exposure and discussed their potential contributions to cold-induced improvements in glucose homeostasis in humans. Here we show that cold exposure has widespread effects on metabolic organs involved in glucose regulation. Nevertheless, cold-induced improvements in glucose homeostasis appear primarily mediated via adaptations within skeletal muscle and presumably white adipose tissue. That's the fat that we wear, right? That's the fat that we don't want to have on our body in general. Brown adipose tissue is different. They're talking about the white fat. So it has a graphic here and it says, okay, cold exposure. And at the bottom, we have improved glucose homeostasis. Okay, but how does that happen? Well, within the fat tissue, obviously there's no shivering going on there. So they have these uncoupling proteins, creatine cycling, fatty acid cycling, improving of glucose uptake, and at the level of the liver, increased insulin sensitivity. And then at the level of the muscles, we have a shivering response, we have a non-shivering response. Ultimately, all those increase glucose uptake within the muscle cells. And then the pancreas itself, they're saying that there's basically a basal insulin secretion and a glucose-induced insulin secretion is also equal. So Seems like, as you can see here, I mean, well, actually, you probably can't see here because it's a very small graphic, and I really couldn't get it much bigger without really degrading the image. But there's a bunch of question marks: question mark here, question mark here, question mark here, question mark here. So there's a lot of question marks. But the bottom line is, when the rubber meets the road, what actually happens to our blood glucose and our A1C, et cetera, like in a clinical setting? Well, again, horribly under-researched, but this is what we got. This is the most recent paper that I could find. It was published in October of 2024, and it says cold exposure alleviates type 2 diabetes mellitus through plasma-derived extracellular vesicles. And it says here that cold thermogenesis or cold thermogenesis mice, let me just make sure I'm CT. So basically they were putting mice in rooms that were 4 to 8 degrees Celsius. And so they called them the CT mice plasma-derived extracellular vesicles remarkably reduced blood glucose levels and improved insulin sensitivity in these type 2 diabetes mellitus mice. This treatment enhanced glucose metabolism, systemic insulin sensitivity, and insulin secretion function while promoting glycogen accumulation in the liver, in the liver and muscle. Additionally, these cold thermogenesis induced extracellular vesicles treatment protected against, basically they had non-diabetic mice that were given this medication that can induce destruction of the pancreas within these mice that causes diabetes, right? They can actually induce diabetes in these mice. So they had another group of mice that were not diabetic, but they were trying to make them diabetic using a very standard lab procedure to make these mice diabetic. And it said that it protected against the pancreatic destruction by inhibiting basically programmed cell death within the pancreatic beta cells, the cells in the pancreas that make insulin. So it protected against these this medication from actually destroying the pancreas of these mice and causing diabetes to begin with. And then it says here that the cold thermogenesis induced extracellular vesicles also shielded the pancreas from destruction and increased expression of these chemicals in fat cells and in liver cells. And in vitro experiments further confirm its pro-insulin sensitivity effects. So unfortunately, at this time in March of 2025, we don't have robust human data showing that your A1C goes from 7.5% to 6.3%. We don't have that. Do I believe that what I'm reading here, the fact that cold in particular preferentially uses glucose and improves glucose and utilization within skeletal muscle and fat cells, 80% of cold is fueled by glucose? Yes, I believe that there's going to be a profound improvement in glycemic control. It's just that we don't have that data yet. And I hope if there's researchers out there that study this stuff or have the ability to do even like some very small trials. I mean, take 5 patients with diabetes, put them in cold water, daily or several times per week for a period of a couple of months, check their A1C on day 1 and A1C at 3 months. I mean, this would not take a lot of money. Okay. This would take very little money and really very little equipment to set up. Let them eat whatever they want. If you want to enhance it, put them on a ketogenic diet and have 2 different, things, but this would not be hard to study. And as a matter of fact, there's a professor, I believe he's of engineering, who owns Morozgo Forge. This could be a study that they could do easily. They own, very fancy cold plunges, very beautiful cold plunges. They could have doctors order their hemoglobin A1C for the starting, and then have them get free cold plunges from Morozgo or from some other, company, and have them do it daily or several times throughout the week for a very standardized protocol. at a very standardized temperature, and maybe even have a couple different control arms. Maybe 5 get 55 degrees Fahrenheit, 5 get 45 degrees Fahrenheit, for a period of X amount of minutes, and you do that for a period of 3 months and see where the A1Cs are at, the fasting blood glucose, the fasting insulins. Although the A1C would be good enough for me. I mean, you get their A1C drops by 50% or not 50%, but by 0.5 or by 1%, then that would be enough for me to say this is an unbelievable treatment modality. It's just that we don't have that data yet. Now, I want to end, okay, on talking about colds been studied on cancer particularly, okay? I put this in here first, okay, because I want to show that there are clinically relevant decreases in blood glucose when it's studied. And that's important for me to show that this is likely going to translate to humans as well. But I'll digress. Before I get to these papers on cold and cancer, let me see what is going on in the chat. Let's start at the top here. Which is more effective, cold water plunge or dry cryotherapy? I don't know the exact number or the physics behind it, but they say that heat transfer between cold water and air is like an order of magnitude. And I have to defer to Dr. Cruz on this one. And he says, he's a huge fan of cold plunging or cold water thermogenesis. I have never heard him say that he's a fan of cryotherapy. So therefore, I'm going to say cold plunging. Let's see here. Is cold shower in the back? Yeah, good question. I think that's to be determined. I think that it could be a start, but I don't think it's going to be where we're looking at. Hey, Doc, thanks for the new vid. You're very welcome, David. I believe cold shower is just as effective. I believe you want to do for 3 minutes. Again, I don't know the answer to that. I think that probably, non-freezing temperatures, we're talking, because the fad is, go into a cold plunge, at 30 degrees or whatever, 30, 35 degrees, get in there for 3 minutes, get my dopamine boost and my norepinephrine boost, and then go out and be productive. That's like the fad, right? What we're talking about is using cold as a way to radically lower your blood glucose and, supercharge your mitochondria, your healthy cells, right? So that would be utilizing a different strategy, completely different strategy. So we're talking about probably warmer temperatures. Dr. Cruz always talks about 55 degrees Fahrenheit being kind of the key, sweet spot of cold, but for longer periods of time. And that's going to take time for people to build up. I'll be very frank with you. I don't tolerate cold very well. When I went down to the cenotes in Mexico, it took some time, but once I got used to them, and they're not as cold as what we're proposing here, that you feel exhilarated, you feel unbelievably good. And so I think it's going to take some time, depending on the person's physiology and makeup and, patience level to build up to where they're able to go. But it may be where we want to do, 55 degrees Fahrenheit for 10 minutes, 15 minutes, 30 minutes. I don't know the answer to these. This is like the edge of science. We don't know the answer, but, and also we're gonna have to design protocols to see, if this person does 55 degrees Fahrenheit for, let's say 10 minutes per day, their A1C went from, let's say 7% to 6.8%. Okay. They do 55 degrees Fahrenheit for 15 minutes per day. It goes from 7.0 to 6.5. There may be a stepwise approach. It may be, you may have to hit a threshold where it then plummets after a certain stimulus. Those are the questions that need to be answered. And those are the questions that I can't answer. But I think that from what I can tell, looking at the literature that we do have, it seems like it's extremely promising. And then when I look at these other rodent studies that I'm about to present on, when we start talking about basically cold's ability to shrink tumors in animal studies, I think that there's going to be a true benefit here. Is a hot 107-degree Fahrenheit sauna then immediately after a cold plunge cryotherapy more effective, like the Japanese practice in Hoxos? So again, those are questions I can't answer because the studies haven't been done. I'll be very frank with you. I think sauna, I'm very bullish on. Pretty much all sauna. I think that the Finnish saunas, the ones that are the hot rocks or whatnot, that get much hotter than an infrared sauna, are very promising when it comes to cardiovascular disease. There's robust data looking at cardiovascular mortality and all kinds of stuff. The infrared saunas, also known as WON therapy within the literature, has some promising studies on cardiovascular, stuff as well. I did do a lecture on sauna, infrared, sauna in particular for, lowering blood glucose. And unfortunately, there is basically no data. And that doesn't mean it doesn't help. It just means there's no data. It's not studied. And it's very frustrating, but I want to make that nuance that I do believe that things like infrared, saunas, probably finished saunas as well, probably lower blood glucose, probably improve A1C. It's just we don't have that literature. And then when you want to, tack on contrast therapy of sauna and cold, do I believe there's a benefit? Absolutely. Is there literature to support that? Not that I've seen, not in this context, at least. The brown fat is mostly on our backs. Should our cold exposure plunge? Yeah, I think so. I mean, basically, I think you should at least try to get the back of your neck, in your cold plunge, if it's possible. With cold exposure, cold temperature will drop dramatically. I'm about to start cold showers along with fasting. Yeah, I think that it's one of those things where you're going to have to be very careful. As a matter of fact, Dr. Cruz, shameless plug, has his cold thermogenesis protocol. It's been online for like 10 years or more. And it starts with face dunking, and then you go into other stuff. It's very comprehensive. I mean, I think that it just makes sense to, at that time in 2013, before fancy cold plunges were available, to take that approach. I think with nowadays with dial, you can set it at 75 or 70 or 72 or 68 or whatever you want to do. You can set the cold plunge at the temperature you want. I think you just lower it slowly. That's what I plan on doing when I get mine. I'll let you know how it goes. What are the areas of the body, the quadriceps, for example, reacting to shivering, the reliable signs of thermogenesis? Yeah, I think that I don't know if we know the answer to that, if you want me to be honest with you. I mean, obviously the bigger muscle groups are going to be more responsible for taking in more glucose. That's why squats are always touted as squats and deadlifts and bench press, right? They're always touted as the compound exercises that have the most muscle fibers engaged and have the most effect on whatever you're trying to do in the gym, right? So the same is going to be for this kind of thing. You want your bigger muscle groups probably to be engaged to get maximal therapy, but also your neck so you can get the fatty acid. Those studies that we talked about, did you see brown fat at all in those studies? I personally did not. I think brown fat has a role here. It's just that it seems as if the researchers believe that it's more the muscles and the shivering response that you get that create the caloric deficit and that, burn all the extra fat and carbohydrates. But I think that probably needs to be elucidated. You saw a lot of question marks on those graphs. I assume the main area would be on the torso. Yeah, I probably tend to agree. That's where the organs are at. How about progressive? Yeah, that's pretty much what I've said. I'm probably late to these questions. I started plunging a year ago. I hated being cold. Me and you both, JP. I haven't started cold plunging. I need to do it. I've concluded that I'm more tolerant to cold now, and I don't need much warm clothes as before. I do plunge at 42 degrees for 3 minutes. Interesting. Yeah, I mean, for general health, that may be enough. I think Dr. Huberman has, podcasts on this and how many minutes per week. But I think that we're looking at it from a very unique perspective of lowering blood glucose as a way to starve tumors. That may require, more time, or maybe it doesn't require more time. It just needs to be elucidated. Do gradual. The aim is to shock the system. The aim is to shock the system, Druid TV, if you're going for the increase in some of the other benefits. I don't know if that's necessarily the case for what we're talking about here. I don't know if it has to be a shock to the system. Uncle Jack says cold affects us. Uncle Jack is talking about Dr. Cruz. Says cold affects us on the physics quantum level via our mitochondria and the inner sun and responding to shivering cold by producing photons and heat. Well, I'm not going to obviously, I'm not going to say that Jack is wrong. I think Jack is right almost most of the time, or probably all the time, although I can't really fact check him because he's up here. But my point is that everything that I'm seeing is that, well, first of all, everything is on the level of quantum physics, right? So it's not like just cold is unique to that. But my point by saying is that we have enough data from the mechanistic standpoint without going to the quantum level, although I would love to go there once I have that knowledge, that it's going to improve mitochondrial function and heat production. In terms of biophotons and stuff like that, I fully agree that probably is happening. It's just that as Dr. Cruz says, until we have photomultipliers at the level of the cell, we really won't know the true light show that's going on inside, right? I believe that we have to turn the tables on cancer. If a chronic condition causes us to be sick, diseased with cancer, we need to attack cancer chronically to lessen its ability to survive. Well, that's kind of what Dr. Seyfried talks about. I don't have the slide pulled up, but if you look at his, it's kind of like a progression of, chronic, press therapies, right? And intermittent pulse therapies until you overcome the disease process. So I can agree with that. I don't think that we're going to do one treatment and be done with cancer. I just don't think that that's realistic in most cases ever, no matter what the technology is. After starving the cancer by lowering GKI, could continued cold further degrade and kill tumors, or a combo of hypo and hyperthermia exposures act as a 1-2 knockout punch? I don't know how cold hypothermia. Well, I'm going to pause what I'm going to say there. I'm just going to get back to the lecture here, because I think that maybe what I'm talking about here will help us understand a little bit more. So, I highlighted, so this paper is titled The Forgotten Art of Cold Therapeutic Properties in Cancer, a Comprehensive Historical Guide, and was published in May of 2023. And it says here, while clinical trials using cold therapy for cancer are still limited or non-existent, you may better say, recent research shows promising results in experimental animal cancer models. The area of research is becoming increasingly significant and warrants further investigation. Will it get done? I would be very surprised, but it should be done. That is true. So this paper is pretty cool because it's showing how cold induces an osmotic imbalance and the formation of these intracellular, extracellular ice crystals, basically kind of like injuring the vasculature to the tumors, as well as having an anti-tumor immune response and leading to apoptosis and necrosis of cancer cells. The next paper I'm going to is titled Brown Fat-Mediated Tumor Suppression by Cold-Altered Global Metabolism. This is the first time we've actually heard of brown fat since talking about cold, although that is believed to be one of the major mechanisms. This is published in Nature, August 2022. So very powerful journal. Most cancers use glycolysis, or the Warburg effect, to harness energy for their own growth, invasion, and metastases. We've known that for 100 years. Activation of thermogenic metabolism and brown adipose tissue, BAT, by cold and drugs instigates blood glucose uptake by adipocytes or fat cells. However, the functional effects of the global metabolic changes associated with brown adipose tissue, brown fat, activation on tumor growth are unclear. Here we show that exposure to tumor-bearing mice to cold conditions markedly inhibited the growth of various types of solid tumors, including clinically untreatable cancers such as pancreatic cancer. Mechanistically, cold-induced brown adipose tissue, brown fat activation, substantially decreases the blood glucose and impedes the glycolysis-based metabolism of cancer cells, the Warburg effect. In a pilot human study, mild cold exposure activates a substantial amount of brown adipose tissue in both healthy humans and a patient with cancer, with mitigated blood glucose uptake in tumor tissue. These findings provide a previously undescribed concept and paradigm for cancer therapy that uses a simple and effective approach. Interesting. So, this is the graphic that goes along with this paper. Let's see if we can follow it. So we have people at 30 degrees Celsius or thermoneutral temperature. This is the picture of a tumor. You can see that it's bigger than this picture. Glucose is being uptaked at its normal increased rate of about 10 to 30x. It's increasing the GLUT1 transporter to help increase glucose uptake into cells. There is tissue hypoxia or pseudohypoxia. There is increased cellular proliferation and growth, uncontrolled growth. We know that. Glucose is being brought in by glucose transporter 1. It's being metabolized to pyruvate. It's going to make ATP through the Warburg effect. It actually says Warburg effect here in this graphic. And then it says lactate, right? We know about lactate, the lactate tumor microenvironment. We know about its inhibitory effects on our immune system, immunotherapy, chemotherapy, etc. We know about all that. You've been a mitochondriac for a while and being part of this channel, you know all about that. you see here that adipose tissue, the white adipose tissue and the brown adipose tissue are in a storage mode. They're not bringing in much glucose. They're basically inert. They're not doing much. You can see that glucose is being much more, taken up by brown adipose tissue because brown adipose tissue has more mitochondria. Those mitochondria are utilizing that glucose. White adipose tissue is a storage vesicle. However, cold temperature, 4 degrees Celsius, we see a decrease in glucose uptake, a decrease in GLUT1 translocation to the cell wall or the cell membrane. We see a decrease in tissue hypoxia, and we see a decrease in cellular proliferation within these cancer cells. We also see a decrease in glucose uptake and pyruvate and lactate and ATP within the tumor cells. We see fatty acids being metabolized, okay? So the white fat, the quote unquote bad fat, is being browned. Okay, they're increasing the amount of mitochondrial number and function, and those browning fat are actually increasing more glucose to themselves, so they can basically cause that shivering, to improve our heat production, right? And then look at this arrow; it goes from larger clearly than the white fat, but we see a giant increase in glucose uptake to this active brown fat, right? Which increases glucose uptake and lowers blood glucose, which ultimately leads to this issue with the cold temperature. Pretty unbelievable and pretty awesome, actually, if you really want to know. This is another paper in Cell Metabolism, October of 2022, starving cancer into submission by activating brown adipose tissue. Basically, I got it mostly for the graphic because the graphic was so cool. So we have like a big tumor on this mice that's like bringing in a ton of glucose. We see that glucose is being diverted to the brown adipose tissue. You see the back of the neck of this mouse, and the tumor shrinks in these mice. And it has basically the same effects as we saw in the last graphic. This is another paper that was published in October 2022 in Cancer Cell, and it has a very similar graphic. We have under warm conditions, we have plenty of glucose going to the tumor cell. It's actually out-competing the normal cells, but when cold is introduced and the brown adipose tissue is being activated, it actually preferentially steals the glucose away from the tumors, leading to a shrinkage of the tumors. The systemic effect on the body, metabolism decreases blood glucose catabolism or breakdown in tumors and arrests tumor progression, offering a novel alternative approach to metabolic-based cancer therapies. So before I end on these last slides, I'll say that I think Dr. Seyfried is brilliant. I think everyone in the lab deserves Nobel Prizes or whatever science-based prizes that are prestigious within the research community. I think that what they have laid down is so pivotal to understand how to enhance what they've already given us, right? They've given us a ketogenic diet. They've given us DAWN. They've given us HBOT. They've given us a framework of how these cancer cells utilize glucose and glutamine preferentially compared to their normal cell counterparts, right? They continue to publish work. I mean, the most recent papers that I saw, at least, were from December, a couple papers in December of 2024, which were unbelievable papers. I have live streams about those papers. But I do think that we have a lot more in our tool bag than we believe we have, okay? Some of those things we've talked about ad nauseum during our glucose series. We've talked about dozens of things that can help lower blood glucose, which can help get you into ketosis better and get your GKI down, which allows you to further enhance metabolic therapy. Those could be supplements, those could be foods, those could be red light therapies, those could be et cetera, et cetera. Now we're talking about cold. Cold happens to be one of those things, okay? Cold can enhance metabolic therapy very likely because it works on the same principles that a ketogenic diet works on, that exercise works on, that red light therapy works on. It enhances metabolic efficiency within our normal cells. It enhances our mitochondria in our normal cells, and it ultimately steals away blood glucose from the tumors. So therefore, they have less available substrate, so they can't metabolize that glucose that they need. And when they don't have the fuel that they need, it doesn't matter if you have a Ferrari or a Honda Civic, or to give a shout out to David, a Shelby Mustang. It doesn't matter if you cut out the fuel to that car and the oxygen to that car, it's going to stop on the side of the road, and you're going to be calling AAA. You're going to be looking for an Uber to get you to the gas station with your gas can, because it is not going to work. Now, I do think that there's other strategies that are not really talked about as fervently as the energy portion of metabolic therapy, that they talk about with ATP. They talk about ATP a lot, with shutting down glucose and glutamine. What they don't talk about much is the oxidative stress component, which I will completely digress on, because we're not talking about oxidative stress. There's other ways we can enhance metabolic therapy aside from lowering blood glucose and lowering blood glutamine. But that being said, this seems to be a very safe, probably uncomfortable, but safe, and probably a very effective way of lowering our blood glucose that doesn't require you to take another supplement, that doesn't require you to go, do something else crazy or take another medication either. Sometimes we use medications to help lower people's glucose. Let me take a look at the comments. Damn, late again. It's okay. It's going to be on replay. I will save my questions because I'm sure they will be addressed, since you are already very deep. I appreciate that. I do try to go deep. We only got what we got, unfortunately, and we don't have what I would be happy with. So if your tumors are in the head or different areas of the body, do those parts have to be exposed to the cold plunge, or is just shivering response that needs to happen? Well, let's think about this logically. We're talking about, your glucose isn't higher in your head or in your leg or in your, right? You have a systemic, glucose. And what we're doing is we're lowering systemic levels of glucose, and we're shunting it away from other parts of the body. And you have to understand that the tumors, the solid tumors at least, they're bringing in glucose 10 to 30x more than the normal tissues that even surround it. They don't even need that much glucose, right? They can live off other substrates like fatty acids and amino acids and ketones and stuff. So no, it doesn't need, you don't need to expose your head to the cold water. I don't believe that needs to be the case. Would you get additional benefit from exposing your head to the cold water? I don't think that it would, but I would be happy to be wrong there. Could you be in a cold plunge with a snorkel, and have your head under the water while the snorkel's above the water and you're getting your entire body immersed? Could that provide additional benefit? I don't know the answer to that. It'd be a cool study to do, but I don't know the answer to that. Let's see here. What impact does it have on blood glutamine lowering, if any? My guess is it's probably minimal. Because my belief, at least before doing all this research, was that the majority of the calories burned were by the brown adipose tissue or the fatty acid, right? Or the brown fat. And, given that, you're not exercising, and it seems to be exercise will lower blood glutamine levels because the muscle cells being worked. I don't know if I have the slide immediately available. I probably do, actually. See if I can find it. This is it. I do have it immediately available. So remember, I don't know if you remember this way back, this slide, that exercise will lower glutamine availability to suppress tumor growth. So the question is that, going back to where we left off, the question is shivering exercise, right? Is shivering exercise? Will that shivering response increase the need for glutamine within those muscle cells? The answer is I don't know. I think the answer could be possibly, but I don't know. If the mechanism was only blood, brown fat, I'd say probably not, have no effect on glutamine. But given that the muscles are so highly involved here, I think it's possible. But I don't know the answer to that yet. All right, going forward, consistent with the changes in this FDG PET scan signal, Brown adipose tissue in cold exposed mice with colorectal tumors had increased levels of glucose transporter 4 and glycotic genes, while tumors had decreased levels of GLUT1, GLUT4, GLUT7, and glycotic genes in comparison to control animals. Basically, the animals that didn't get cold, they had the same amount of glucose transporters in the cancer cells and glycolysis-related genes. However, the people who the mice that got cold that had colon cancer, they had decreased levels of those. The redistribution in glucose uptake and catabolism between activated brown adipose tissue and tumors were underlined by decreased levels of blood glucose and improved insulin sensitivity. Again, it's a systemic response. So it just goes to show you don't have to be exposing that part of the body that has a tumor to cold. Now, will that enhance it? Maybe. Don't know the answer to that. Accordingly, tumor progression was significantly reduced in xenograft models in human colorectal cancer and pancreatic ductal adenocarcinoma, a very common cause, very common pancreatic cancer, an allograft of murine, colorectal cancer, melanoma, pancreatic adenocarcinoma, fibrosarcoma, and breast cancers, and in spontaneous MMTV PYMT model. I'm not even sure what that is. I have to look that up. And APC colorectal models. Consistent with the role of uncoupling protein 1 in brown adipose tissue activation, whole body genetic deletion of UCP1 ablated the increase in glucose uptake in brown adipose tissue and decrease in blood glucose in colorectal tumors and prevented the effective cold exposure in tumor tissues. So that actually is kind of controversial, right? So basically, all the studies we looked at before these mouse cancer studies was saying that it was all the shivering response that gave us the benefit of lowering blood glucose, right? However, someone had asked, are there other ways of executing shivering besides cold temperatures? I don't know about shivering, but there are other ways of enhancing uncoupling protein 1. That I can say there are. Supplements, foods, etc. I think even exercise increases uncoupling protein 1. I have to look to be sure. I don't want to give false information. My point is that when they looked at animal studies for cancer particularly, and the cancer's ability and cold's ability to steal glucose away from cancer cells and basically like shrink these tumors, what they did was they tried to prove it that it was only the brown fat that made the biggest difference. And they did it. Okay. The way they did it was they took these mice, made them cold, and they then deleted this uncoupling protein 1. And that like completely mitigated the improvements in blood glucose response when these mice were cold and in the tumor cells. So it stands to reason that brown adipose tissue seems to be at least as important, if not more important, than muscle shivering for lowering blood glucose and sealing away from tumors. We don't know for sure because we don't have enough studies. But what I'd say is that brown adipose tissue seems to be critical for the stealing away of blood glucose away from tumor cells, at least in animal models, probably in humans as well. It's just that we don't know yet. Wim Hof holds the world record for swim time under ice. I know, the guy's unbelievable. He also climbed the highest mountains. Yeah, the guy is like an alien. Like, he's unbelievable what he can do in the cold. I mean, I know. Going to a cold room, the same thing. No, it's not. It's not the same thing going to a cold room because air transfer of heat is much different than in water. I don't know the exact figures, but it's significant. It's like orders of magnitude different. Later for the snorkel, holding your breath would be far more stressful, thus for more beneficial. Well, define stress and being beneficial. I mean, if it's a catecholamine response and a cortisol response, that potentially could increase blood glucose, right? Because that's what they do. So I don't know if that's necessarily true or not, but I think we're really out on the limb speculating about snorkels and stuff like that anyway. So I think let's just wait for more data to come out. What I mean, maybe cancer consumes glucose in huge amounts at the same time absorbs poison, which kills him. I don't know. Maybe I'm a physicist. Well, I am not a physicist, so I'm sure there are many things that you know that I don't know. Atoms, stuff like that, not medicine. That I understand. Well, it's not like they're disconnected. They're just disconnected in real life and in medical literature and training. It's not that they're disconnected in you know what I'm getting at. Be careful of magnesium chelate with glycine. After a month and a half, my breast cancer tumor ballooned. Then I accidentally burned myself with iodine, DMSO, and IR sauna. Cold showers sound perfect. I'm really sorry to hear about your breast cancer getting worse. So is it your belief that you believe that the magnesium was the only reason for it? Magnesium glycinate was the only reason for it? Or do you think there's maybe other factors involved? Because I also believe there could be a problem with magnesium glycinate, but I don't know if I would think it would be that much of an effect, but man, if it is, that means that we need like an emergency broadcast with like magnesium glycinate in cancer patients then. L-proline is bad for cancer. Well, let's be very honest, right? Like in order for a cancer cell to grow, it needs substrate. It needs all amino acids, right? It needs methionine in high amounts, which we're going to have a whole series on because I've been how do I want to say it? Motivated by one particular viewer. If you're watching it, you know who it is, to do a deep dive on the methionine and the Hoffman effect and stuff like that. But let's be honest, the cancer cells tend to need all the amino acids. They're going to need fatty acids. They're going to need carbohydrates of different types. And the reason is because not that they're using those things as fuel. Okay, the engines aren't using those as fuel. What they're using is that to grow and proliferate and to make new proteins and to make new enzymes and to make new cell walls and cell and membranes and organelles and stuff like that. And those require proteins, right? So they're going to need all the amino acids. It's just that the bottom line is that those cancer cells, they rely completely on glutamine, right? So glutamine's a special one. It's above and beyond the rest, you know? And so therefore, yes, proline probably is bad for cancer. You know, it's just that I don't think anybody's recommending a low proline particular diet. You know what I mean? At this point, in general, a therapeutic ketogenic diet is going to be a low protein diet by theory, by definition. You're not going to be getting massive amounts of protein. Now, we don't want you to be underdoing protein either, you know, but at the same time, it's not a massive amount of protein you're doing. So it's going to already restrict some of the amino acids coming into the body, right? And then, of course, there's amino acids that our body makes on their own, the non-essential amino acids like glutamine, which there's no dietary strategy that can really help us there, right? Now, maybe what you could say is don't take amino acid supplements. You know, that would probably be a good idea, you know, when it comes to cancer. Okay, let me finish out this bad boy with this last slide, which is, again, a very similar graphic, but it's just a neat graphic shown in different ways. So this is the last paper I'm going to show. I don't know why it's so small, but it is what it is. I should have made it bigger. I'm going to read it off for you, okay? It's called Cold Snap for Cancer. Cold-induced boron thermogenesis starves tumor growth. Published in 2023, January 9th of 2023. In a recent study published in Nature, reported that cold-induced thermogenesis, brown fat activation, or cold-induced thermogenesis, impedes glycolysis and reduces tumor growth in multiple cancers, depening on deepening our understanding of the complex role of fat metabolism in cancer. It later says, well, actually, I'm just going to read the whole thing. It's not very big. Cancer cells switch energy generation from effective Krebs cycle and oxidative phosphorylation to inefficient aerobic glycolysis, a phenomenon known as the Warburg effect. Such adaptation dramatically increases glucose uptake rate, helping tumor cells overcome the energy shortage and providing a carbon source for anabolic processes. Approaches to cut off the glucose supply from tumor microenvironment, TME, such as calorie-restricted diet, fasting, metformin, showed promising results in controlling tumor growth. In the Nature study, found that immunocompetent mice simultaneously implanted with colorectal cancer cells experienced marked tumor suppression when mice were housed in 4 degrees Celsius environment. Now, again, these mice studies that were done with cold air. Okay, just be very frank. But given that what we know about cold water immersion, you're going to have a much better heat transfer and probably more effective with doing cold water immersion. But they're not putting these mice in cold water, just to be very upfront. I forgot to mention that. With a striking 80% reduction in tumor growth after 20 days. 20 days, the tumor reduced by 80%. Cold acclimatization led to an improved survival of mice and significantly reduced tumor hypoxia, angiogenesis, or making of new blood vessels, and tumor cell proliferation or growth. So, does this jive with what Siegfried is talking about? Yes, it does. It lowers blood glucose availability to tumor cells. So therefore, it completely takes advantage of the metabolic theory of cancer. Does it show extreme promise when it comes to lowering tumors' ability to grow? Well, in a mice study, it lowered the tumor volume and proliferation by 80%. Does this need to be studied more before it's ready for prime time and we're able to make broad claims? Yes. Do many people who are listening to this lecture, podcast, have the ability to wait 5, 10, 15, or longer years for those studies to come out if they get done at all? No. Do I believe that this could be very helpful to enhance metabolic therapy? Absolutely. If you like videos like this, please like, share, and subscribe. I have a lot more to share. We're going to be getting into glycolysis, and we're going to be getting into things that can decrease glucose uptake into cells, as well as glucose utilization into cells by blocking glycolysis, by blocking lactate dehydrogenase, and even hypoxia factor 1 alpha as a way to cut off the Warburg effect at the knees. So until next time.